Hey, welcome to C4.1 Populations and Communities. We have the SL guide here. It's quite lengthy. More SL, there's no HL to this section, so everyone needs to know it. Okay, a few ideas how you can use the information in the video to get a grade 7. Just watching videos is not going to get you a grade 7. You must do something with the information. Key terminology, you can see it's all green because it's all SL, lots of key terms. This seems like it should be a relatively straightforward topic, and yes, the content is relatively straightforward, but it's quite wordy, and there might be quite a lot of terminology you haven't heard of before. Okay, let's make a start. So we've got estimating population size. So we need ways in which we can estimate the size of a population without counting every single individual within a population, because that's just not possible. So we can use quadrats and transects. You've probably met these before in previous study if you did GCSE or MYP. With quadrats, you can sample sessile organisms. Now, sessile organisms are organisms that aren't going to move very much or at all. So plants, for example. And quadrats are used for random sampling. So we're taking a random sample, we're removing bias by using random sampling, and we will estimate the population from that. So what we do is we place the quadrats in random positions, but the positions have to be determined using a random number generator. We are not standing in a field and throwing a quadrat. That is not random sampling, because where you stand has not been decided randomly. You need to calculate the average in 20 quadrats and then multiply by the total number of quadrats in the whole area. So you need some sort of method of calculating the population in that area. Transects. Transects are a line that go from one place to another. Now you can have a line transect or a belt transect. A line transect is literally just a line. A belt transect is where you have an area in between two lines that you're going to sample. Again, we're going to sample sessile organisms, so ones that aren't going to move very much, but this is going to be systematic sampling. So we're going to sample at specific points along that transect. We'll place the quadrat at the specific intervals along the line, so we'll decide what the intervals could be. They could be 1 metre intervals, they could be 10 metre intervals, they could be 100 metre intervals, depends on the area. And you will, ideally, your transect should go from one area to another area where there will be some sort of change. So from the water along to a dry area, or from an urban area to a rural area. I've said that are useful on rocky shores or urban to rural areas. We've also got the mark release recapture method. This estimates the size of a population in an area. The first thing that you have to do is you have to catch a certain number of the organisms within a population, of the individuals. So you might choose to catch 100. You then need to mark them, but you have to ensure that the marking does not cause harm or affect the chances of survival of the individual. So for example, if you're going to catch snails, you cannot paint them with neon yellow nail polish in order to mark them, because A, that might be toxic, and B, that might make them more obvious to predators and therefore more likely to be eaten. You also must make sure that you mark them with something that isn't going to come off. So the marking is going to stay on until the next time you come back to find them. Because once you've marked them, you're going to release them. Then you have to decide on a set amount of time. So it could be a week, a month, it could be a year. And then you're going to use this Lincoln index to estimate the population size. So we have the number in the first capture times the number in the second capture over the number marked in the second capture. So when you capture them a second time, you need to count how many you're capturing, and then you need to count how many of your snails are marked with the neon nail polish, and then you put it into the Lincoln index, you do the calculations. There's some assumptions we need to make, so we have to assume that there's no immigration or emigration from the population, and that all organisms have an equal chance of survival. I also said that the marks need to stay on during the time of the investigation, and the markings cannot harm the animal. This is the population growth curve, so we're going to have a look at what happens in the different phases of population growth. First, we've got the lag phase, where we can see that the population is low in number but steady, and then it just starts to rise. Then we've got the exponential growth phase, which we can see there's a huge increase in population Can also be called the logarithmic phase because it's usually on a logarithmic scale, rapid increase in the population size. We've got the transitional phase. At this point, the growth slows down, but the population is still increasing. So it's not this rapid growth, it's slightly slower growth until we get to the stationary or plateau phase. So we can see that the population is high and it's reached carrying capacity. Carrying capacity is the maximum number of organisms that the habitat can support. There's also fluctuations here, so fluctuations as a result usually of predator-prey relationships, which we'll see very soon. There's limiting factors that could prevent population growth, so things like lack of oxygen for respiration, disease might wipe out a number of a population, there might be many predators, which means that the population can't grow, sunlight availability for plants, they can't photosynthesize at a maximum rate, or food availability as well. We've also got density-dependent factors and density-independent factors that can affect the population size and population growth. So density-dependent factors are self-explanatory, so they're dependent on how many of the population is in the area. So for example, disease would spread rapidly in an area that's very dense with the population, whereas if it's not very dense, then the disease wouldn't spread as rapidly. Density-dependent factors keep the population below the carrying capacity, so it doesn't let the population growth increase to that carrying capacity point. Density-independent factors are usually abiotic factors that affect a population regardless of the density. So natural disasters would wipe out a huge number of the population and affect population growth. Climate change also might affect population growth, and there's more about that in the climate change section of the spec. We've then got positive feedback and negative feedback, which you probably have met before. Positive feedback is where something causes the population to increase, and negative feedback is something that causes the population to decrease. So that could be food availability, it could be competition, it could be a disease, but whatever it's doing, it's causing the population to decrease. Okay, we're going to have a look at some of the relationships between different organisms. So we can have intraspecific or interspecific relationships. Intraspecific relationships are relationships between organisms of the same species. An intraspecific cooperation is where some organisms will help others within their species to ensure survival and to ensure that young are born and that the species can continue. Intraspecific competition is where members of the same species might be competing with each other for the same resources, for example, a food source, because they're occupying the same niche within the ecosystem. We've also got interspecific relationships, so these are relationships between different species. We've got herbivory, so this is where organisms consume plants which damages or kills them, and that would be consumers that are consuming the plants, obviously. Predation, because organisms will eat each other, or consumers will usually eat other consumers or plants. Interspecific competition, which we said is between different species competing for the same resource. We've also got mutualism, so this is where two species are providing one another with something that both benefits them, and we'll look at an example of this in a minute. Parasitism is where one species lives and depends on the host, so an example could be like a tapeworm, which lives inside the digestive system and depends on the human to provide them with nutrients. And then we've got pathogenicity, and that's the fact that pathogens can cause disease in other species, which obviously we know a lot about from the disease sections of the spec. Okay, we've got mutualism up here. So we're going to talk about root nodules. Now we're looking at this diagram of the nitrogen cycle just here. So we can see that this is a plant, and this is what we call a leguminous plant. So any plants essentially that grow beans. Now what they have in their roots are these little nodules, and the nodules contain a bacterium called rhizobium. And what the rhizobium are able to do is they can take nitrogen from the atmosphere and they can fix it into nitrogen containing compounds that the plants can use. If the plant is not a leguminous plant, then within the nitrogen cycle, the nitrogen from the atmosphere has to go all through this process in order to become nitrates that other plants can then take up. So these leguminous plants are at a really good advantage because they have these rhizobium in their root nodules. And we say that relationship is symbiotic, because they're both benefiting. The plant's benefiting from that, and obviously the bacteria are benefiting from that because they've got somewhere to live, and they've got nutrients as well. We've also got mutualism, where both species provide each other with something that benefits them. Mutualism in mycorrhizae, so some plants have a network of mycorrhizae within the roots, and mycorrhizae is a fungus that lives within the roots. You can see here the difference, and what it does is its system of hyphae increase the surface area for the absorption of nutrients, and the mycorrhizae then give some of those nutrients to the plant, so the plant is able to take on more nutrients with the mycorrhizae than it would be without. So the plant gets more nutrients, the mycorrhizae get nutrients, and somewhere safe to live. Okay, the final example of mutualism is this zooxanthellae. I think that's how you say it. And this is all about coral. So coral polyps are teeny tiny animals that live that build the coral reefs, and they have tentacles which contain photosynthetic algae, which is a type of dinoflagellate referred to as this zooxanthellae. And the dinoflagellates provide food for the coral polyps as a result of their photosynthesis. So they photosynthesize, they make carbon-containing compounds, and then the coral polyps can use these. The coral polyps provide a home for the dinoflagellate, so therefore it's a symbiotic relationship or a mutualistic relationship. And the dinoflagellates are actually really colourful organisms, and they're responsible for the bright colours of the coral reef. And if they die, then the coral will look white in colour, and that's known as coral bleaching, which you'll see again in other areas of the spec. So another example of mutualism that the IB want you to know about. Okay, we then move on to endemic and invasive species. So endemic species are species that are found only in that specific area. They're not found anywhere else. What sometimes happens is other species are introduced to an area in order to control endemic species. And if these invasive species don't have predators, they're able to outcompete the endemic species. It means that their population can increase rapidly. And that disrupts the food webs and the ecosystem within the area that they've been introduced into. And I think the IB would like you to know an example of an invasive species from your local area. One example is the cane toads in Australia, if you want to look that one up. Okay, we then have studying and being able to investigate interspecific competition. It's possible to develop original methods for testing for competition and to see whether species are competing with each other. So sometimes the species might not be in the same area together because one has outcompeted the other, and investigations often put the results into a table called a presence-absence matrix, and you will see that on the next slide. This allows us to see whether patterns exist and whether there has been any outcompeting between species. So within lab investigations, you can grow seeds outside of their natural habitat. You can then put different species together to see which one is going to survive. You can allow them to grow separately to look at the difference between their whether they thrive with another species or without another species. We've got field investigations, which is where researchers manipulate the natural environment. So they go to where these plants actually live and they manipulate the area there. You can add or remove specific compounds in soil, change other variables to affect the plant growth. And then we've got field manipulation, where researchers can add variables and specific species to investigate if or how they affect the other species. So there are ways that you can test for interspecific competition. They might give you an example and you might have to design a method within the exam to test for interspecific competition. Now we're going to look at the chi-squared test, which is a stats test used to compare observed and expected values. So the observed values are the values that you collect within your investigation, and the expected values we're going to calculate using your presence and absence matrix. We talked about this on the previous slide, that's just here. The first thing we need to do in the chi-squared test is write your null hypothesis. Now the null hypothesis always states that there will be no significant difference between the observed and expected values. We then need to determine the expected frequencies. So your observed frequencies are here and the expected frequencies are here. So we've filled in the presence-absence matrix, and now we're going to calculate the expected values. You can see that we've got this total here and this total here. So we do this one times this one divided by 150, which is the total there, in order to find our expected values. So expected values and observed values that we counted. We're then going to calculate the chi-squared values. So you can see that we've got observed values, expected values. We then do observed minus expected squared over expected, because this is your chi-squared formula. And once we've done that, we add them all together because it's a sum of, and the chi-squared value is just down here. We then need to calculate the degrees of freedom. The degrees of freedom in chi-squared is always n minus 1. Now, n is the number of possible outcomes, which in this case is 2, present or absent. So therefore, 2 minus 1 is 1 degree of freedom. The next step is to determine the critical value. Now, we have to use the critical values table in order to do this, and we're always going to use a significance level of 5% or 0.05. So we look at for the 0.05 column, and we look at the degrees of freedom here. We can see 1 degree of freedom and 0.05 is 3.841. We then have to compare our chi-squared value that we found here with the critical value which we found here. And we can see this is the interpretation of our test here, that the chi-squared value is smaller than the critical value. Therefore, we accept the null hypothesis, and we can state that there is no significant difference between the observed and expected values. This might be useful for your IA. You may have to calculate or interpret chi-squared within the exam. We're going to finish off with looking at some methods of controlling the number of specific populations. So the first natural method of control is predator-prey relationships. We can see that the orange line are the wolves and the blue line are the rabbits. So the wolves would obviously eat the rabbit. Now we can see that as the population of wolves increases, shortly after that, the population of rabbits is going to decrease as well. And then as the population of rabbits increases, then the population of the wolves increases just after that, because they've got more food source. So this is an example of density dependent population control. We've also got top-down and bottom-up controls. So all limiting factors can be either top-down or bottom-up, and top-down controls are controls that reduce the population size as a result of predation. So if you increase the predators, more of the prey will be eaten, and that will decrease the population size. And bottom-up controls reduce the population size as a result of lack of resources such as food or minerals. So if for some reason a disease wipes out a specific crop, then that might impact whichever consumer eats that crop, and therefore their population size would decrease. And bottom-up controls are common in aquatic ecosystems. Okay, and then we move on finally to metabolites. We have primary metabolites, which are the molecules needed for the basic functions of life, and then we have secondary metabolites, which are molecules that can be released that have a negative impact on the species living in close proximity to the original species, and this process is referred to as allelopathy. In plants, some plants can release molecules that inhibit the growth of neighbouring plant species, which is handy because it means that if other plant species can't live close by, they won't use the nutrients within the soil, so that original plant can use all of those nutrients. And then a really nice example is the fact that some fungi release antibiotics such as penicillin. So if the fungus that releases penicillin is growing on an agar plate, it means that no bacteria will grow around that fungus. So it can then take all of the nutrients from the agar for itself. Two nice examples of allelopathy that the IB want you to know.